And when I went to the agricultural school and talked to them about coming there, and they said, you're the first physician that has come to us to actually collaborate, besides one other person, and nobody's done it from dermatology standpoint. So I do a lot of just general medicine, general dermatology, infectious disease, surgeries, and everything, but this is the area I think is very interesting because we can make a bigger difference in people's lives. So anyway, if you want to come out to Davis, it's a lovely area. A lot of agriculture is going on. We also have the veterinary school, so a lot of research on milk, animals as well. So breast milk, that's another area. So let me get right into what we're going to talk about today. This is overall kind of an overview of what we'll be talking about. Take you through it in about 30 minutes. So first we'll talk about the medicinal benefits of plants. You may all know many of these things, but it's nice to review some of this. Then I'll show you some of the studies we're doing with the cells. I'm very interested in I have my own lab with cell culture, and we're looking at how the cells can be modulated with these plant properties. Then eventually I do a lot of clinical trials too. In fact, I've set up a couple clinical trials right now. We can take what we find in the lab and then immediately apply it to human use. The beauty in skin is you can see what you're doing, and also the safety profile is a little bit easier to understand. So I don't have to do animal studies necessarily if we can show it's already a food. If it's already a food, the safety is not that much of a concern. People have been eating it for thousands of years. If I'm just applying it for some other principle, then we're able to take those to clinical studies. And then finally, some thoughts about areas that I'm really interested in. I was hoping that I could collaborate with some of you. And for some of you that are students, I really hope this is at least in some way makes you think a little bit more about the possibilities of the education that you're getting here. I think it's a fabulous institution. Just coming in, I can tell people here are really passionate. So all of you that are here are very lucky to be studying at this institution. So FDA has approved many drugs, and there are many drugs that exist from plants. This, you may be aware, aspirin from willow trees. This is huge. We use it all the time. And then Taxol is another one that was discovered in North Carolina when they first were trying to figure out new ways of treating cancer. And so now it's just pretty much wholly produced from the plant. They have synthetic pathways. And this one came out in January 2012. There it is, species, Peplis, Euphorbia peplis. So this is originally was a weed that a lot of the Aborigines in New Zealand and in Australia were putting on their face to help them with their precancers. Then they isolated the compound. It's a triterpenoid, and it became FDA approved just in 2012, and it massively reduced the time of treatment. So people are starting to understand plants are not just novelties; they actually have true potential. These are how some of the phytochemicals are available. The FDA does not regulate any of this as long as you don't claim that they're going to treat a disease. If you say they're going to treat acne or treat eczema, they will come after you and fine you heavily. But if you say it'll make your wrinkles look nicer, it'll make your oils not produce as much, or for eczema, you won't itch as much, that's fine. Because you're not treating a disease, you're treating a symptom on the skin. And so that's okay. So they come in foods, plant-based extracts. Here you can see just curcumin. God only knows how they got the curcumin. Nobody knows. Sometimes these people, they'll make extracts and you don't know how they made the extract, what part of the plant they took the extract from. And sometimes they get adulterated too, which is another issue. But for example, this is the cosmetic industry. Magic white ice. You know what's in magic white ice? There's a lot that's in magic white ice. So much that I don't even know what's in magic white ice. You can see there's this extract, that extract, this extract, this extract, this extract. Who knows how they made the extracts? You know why they put all these extracts in? Because it means they can charge more money when they actually take it to the consumer. But nobody knows if it's actually effective. And in fact, this rigorous research on a lot of extracts is lacking. I'm writing a textbook called Cosmeceuticals and Active Cosmetics. The whole principle is you take any natural product and you should expect people to do research on them so that you have a higher level of evidence to show that it actually has an effect. People will claim anything, but the question we always ask is that true? And what was your research model that you used? Because now, when you come to the marketplace, if some people are starting to do that, the people that don't provide evidence, they're at a huge disadvantage because people are always going to choose the product that has more evidence behind it. So when they put all these things in, of course it has to feel good. If Magic White Ice feels better than any other product that has evidence, they'll still buy Magic White Ice. So there's always marketing. So different phytochemical subgroups. This many of you are aware, but just so that you know some of the things that the general population is really interested in. Flavonoids, they tend to have these conjugated structures so that they can be good receptors for free radicals. So excellent antioxidants. We now know, and I'm not showing this, but we've done this research, that they're not just passive antioxidants. Some of these can actually stimulate your cells to have heightened defense mechanisms. So previously it used to be thought, oh, we can just put this in, it's an antioxidant, but now we know that it's more than that. Some of these can do more than that. They can actually prime your system to become more able to take on some sort of oxidative stress, even in the absence of this being there. So this is what is called a cosmeceutical, because now it's got an active property to it, but it's also still used in a cosmetic sense or in some sort of cosmetic line. Cosmetic doesn't necessarily mean non-medicinal. It just means the FDA doesn't review it. So. Then we have the terpenes and terpenoids. Again, originally, this was the aromatic portion of the plant, the essential oils. This was thought to be what would give a nice scent to things. One of the research areas that we're looking at in Davis is one of the people I'm collaborating with. I'm not working on it, but how do plants communicate with each other? How do they tell each other that there's some danger in the area? How do they tell each other, oh, we have good growth conditions? One of the thoughts is some of these terpenes are part of that signaling messenger system. And they're concentrated in the volatile portion of the plant, so the essential oil. And if you peel the skin off of your fruits, you're removing a lot of these. Just remember, it's in the skin. So I'm not going to eat the pomegranate skin, but the apple skin I'll probably wash and eat. So just something to think about. And when you look at human health, a lot of anti-inflammatory or pro-inflammatory, depending on what you take in, and then it modulates the immune system. People ask me all the time, doesn't natural mean it's good for you? Poison ivy is natural. I don't think it's good for you. If you put poison ivy, I don't know if they have poison ivy here in India much, but if you put poison ivy on your skin, you'll blister all over the place. So natural doesn't necessarily mean good. It just means it's natural. Then you have other subgroups like the anthraquinones. This is what gives a lot of fruits and vegetables that are colored. But one beneficial area is we've started to realize this can be actually a natural sunscreen. Ultraviolet light, visible light, and one of the examples is right here. But the whole idea is that they can also have other properties, anti-neoplastic. But they're an aromatic group, so they absorb UV light very well. So this is an area where now a lot of companies are looking at natural sunscreens and ways that you don't put chemicals in the body. The reason is, when people put chemicals on their body, they've noticed, you probably haven't heard this from any sunscreen company, but I'm going to tell you. When you put on chemical sunscreens, they've done this in people. The sunscreen absorbs through the skin, and people have measured it in the urine, which means it's getting into your bloodstream. People will say, oh, the amount that you get in is not that high, but it probably will embed into your fat, because these are all fat soluble. So what you measure in the blood is not totally what you're absorbing. And they have estrogenic properties sometimes, and they're shown in some women that use a lot of sunscreen. They're getting conditions like endometriosis that are related to a high estrogen in the body. So is the chemical sunscreen totally safe for you? The sunscreen company will say yes. A lot of us will say we don't know. We're not totally sure. Doesn't mean we shouldn't use sunscreen. It just means we need to do better. We need to have a better way of approaching these things. So different parts of the plants are important. This is another important concept. The root is different from the flower, different from the stem, different from the leaf, because they all have different functions. If you go into the Ayurvedic literature, I studied in Ayurveda a little bit as well. I'll show you, Ayurveda was initially thought to be very empirical. Now the research is starting to show from Western medicine there's a lot of overlap between Ayurvedic medicine and allopathic medicine. And I say this as a fully trained, fully embedded allopathic physician, that Ayurveda actually wasn't that wrong. In fact, I think they got a lot of things right that we still have yet to figure out. So, but one of the things is each area of the plant is unique, and for example, in pomegranates, if you take the actual fruit, that's not where a lot of the ellagic acid and things are located. It's actually in the peel that people are throwing away. So that may even be a business opportunity for anybody who does things. This is some research that we did. We were looking at specifically for wound healing, and we know that these are the plants that are that have been mentioned for wound healing. And what we showed is if you take the solvent of extraction and look at the antioxidant properties, the solvent matters a lot. And not only that, it differs for each plant. So if you look at the roots, for example, ginger, turmeric, it's very interesting because as you move towards something that's less aqueous, you actually get higher antioxidant properties, which may make sense because in the root, doesn't tend to be flavonoids. It tends to be things like the curcuminoids. But then when you have leaves like neem, echinacea, aloe vera is a unique one. Aloe vera actually went the other direction. But the whole point here is that you really, for every plant, need to test the solvents first before you start thinking about what are the next things to do. Also, this is just antioxidant property. A lot of plant science has just focused on antioxidant property, but there's a lot of other properties and other bioassays that have nothing to do with antioxidant status. So if you look at this and say, oh, look, ginger is not as good as echinacea, well, that may not be true, because if you look at anti-inflammatory, that may have nothing to do with antioxidant property. So you always have to do the assay that is relevant to whatever you want to test. So that's the other aspect that we've been looking at. So the difference between phytochemicals and plant extracts, which again you may be well aware, when you have a specific compound, you can focus on just what that specific compound is doing and what is the effect, and you can patent it. So this is very lucrative for a pharmaceutical company. But what you miss out is when you have mixtures of compounds, and I mentioned the FDA just approved that mix for warts. I think what they have realized is that sometimes when you have a mixture of compounds, they have synergistic effects. One may be more antioxidant, one may be more anti-inflammatory, one may be more antimicrobial or antiviral. And so it's not easily patentable, but still you can patent it if you have the proper extraction technique or a proper formulation. But this is also the new concept is moving towards this, where it's not single target, single molecule. If you ever come to Sacramento, I don't know if you're aware, when you give presentations, 15 minutes in, people start to think about what they're going to do for dinner, what they're going to do for lunch. It's called float time. I don't know if you've ever taken a course on public speaking, but it's true. If I'm boring you, that starts to take effect about 15 minutes in, so you have to break up the boredom. That's why I'm showing a picture of Sacramento here. So we have a nice river, nice bridge, very nice area. You should come and visit. It's very pretty. It's not as humid though. It's very hot and dry. But we have good agriculture there. And then let me now talk to you about some of the studies we're doing with sebocytes. So sebocytes, this is the skin in cross section. So it's, if you just look at your hand, you'll see there's small little hairs on your arm. In this hair, you'll see there's a sebaceous gland. This is where the sebocytes sit. This is the epidermis, which is your protective layer of skin. And underneath is the dermis, where you have the blood vessels and the sweat glands and also the nerves. But the sebocytes are very important. They secrete this oil substance that comes up, it's called sebum, all the way to the top and coats the very top of the skin. And this is what helps create the barrier for your skin. And this barrier is important. If you don't have a good barrier, we all die. It's actually just 15 microns thick. And that's created by the epidermis, and then the sebaceous gland augments that. This is the only lipid-producing cell in the entire skin. I mean, the epidermis produces some lipids, but not to the same level as the sebaceous gland. Also, sebaceous gland is in direct connection with blood vessels, whereas here, there are no blood vessels after you get into the epidermis. So everything is through diffusion to get nutrients. But here, you can directly interact with the blood lipids, the blood hormones, and so whatever your internal nutritional status is, one of our goals is to understand, can we actually catch that through the sebaceous gland function? So can you then back out, if you can see some certain changes in the sebum, can that help predict where your nutrition status is? We don't know, but this is a brand new area of research that we're really kind of interested in. I'll talk about acne. It's a disease that affects almost everyone. Everybody's had some little bit of acne, some people lucky not to get acne. This is a disorder of this sebaceous gland unit. And one of the things with acne is, although it's not life-threatening, sometimes the psychosocial, psychological effects are lifelong. Because if you have scarring or redness and you don't develop the confidence when you're young, It's harder to develop that confidence when you go out into the world. You know, the way you interview changes, the way you behave changes. You may say that's not that important, but I will tell you, if you look at people and you see who are the really confident people who get the jobs, who get, you know, apply to medical school, you go to an interview. You apply to law school, you go to an interview. You apply to business school, you go to an interview. The people that can interview well always will get the job. It doesn't matter, you know, qualifications get you to some level, but your ability to project yourself makes a huge difference in how other people will take you and how seriously they'll take you. Sometimes when you have acne, and some of these children, they're so focused on this, they don't get good marks in school, and people say they're stupid. Then we fix their acne, then they come back and they're getting straight A's. So were they really stupid? No, I think what it is they were mentally, they weren't ready to. So it's a big area. I want to point out something. This is huge. It just came out in Science in 2009. This is a whole new way of thinking about your skin. So what it says is your skin is not the same everywhere. The skin on the face is different from the skin on your hand, different from the skin on your chest, different from the skin on your legs. This is why certain diseases like acne only affect the face, chest, and back, but then other diseases may affect the legs. So the idea behind it is you have unique microbes on every single portion of your skin. For example, the sebaceous areas where you have a lot of active sebaceous glands, those oil-producing glands, face, armpits, chest, the groin area, basically all the folds, you see more of this bacteria. So what they did is they actually look at the relative abundance of every bacteria. And what you see is Propioni bacterium acne species is relatively abundant in the areas, in every single human being, regardless of whether you have acne or not. Then when you go to areas that are more wet, so if you, some people in the areas, skin folds, if your feet are always wet, they get an abundance of Corynebacterium. And then if you have more dry skin, then you get a different microbe dominance. The point here is our skin is different in every single location. So if we can adjust how we deliver some plant chemicals to address the particular pathogen in that area of the body, then you can do a better job of controlling diseases. So we have to be aware that there are different pathogens. But not only that, in disease, these pathogens actually change. So if you look at things like eczema, and then they looked at the microbiome, what they found, this is overall right here. When someone doesn't have eczema, do you see there's not as much pink in the control down here? When they don't have eczema, they don't have staphylococcus. That's actually not normally found on our body, Staphylococcus aureus. When you have eczema, but it's controlled, you have a little bit of staphylococcus. But see, when you flare, the staphylococcus is, what happens is there's something called the Shannon diversity, which is how diverse are your bacteria on your body. That diversity goes down, meaning you have one bacteria that becomes dominant. So in a flare, you'll actually have this dominant bacteria. So if we could use a phytochemical instead of antibiotics to address this, then that would make a huge difference for treating people's eczema. So this is where, normally we give antibiotics, but if you keep giving antibiotics, at some point this bacteria will become resistant to that antibiotic. And that's actually a huge public health problem. Already we're starting to see resistance, and then what you have is resistant bacteria, and then you have to come up with different antibiotics, then you have the side effects of those antibiotics. So if in a cream we could actually put some sort of phytochemical that is directed towards this, then you can control their flare without exposing them to steroids or antibiotics, and so it becomes a huge way of treating these folks. So to get into one of the phytochemicals that I got quite interested in, there was actually the research came out of India when you're looking at Propioni bacterium acnes. That was the one area that I was looking at. There's a few that they studied, but they found that of these, certain of the bacteria actually had the ability to inhibit some of the P. acnes induced oxidative stress. One of them was rubia. This is actually something that was predicted in the Ayurvedic literature, but we didn't do much work in the allopathic literature. Of course, there's the other classic ones like turmeric and neem and aloe vera. There's the other classic ones, but this one kind of jumped out. So what we did, and then this is just to show that they looked at the reactive oxygen species production in these neutrophils, which are a certain immune cell in the body. And they found certain of these really inhibited the inflammatory response. So what we did was we took things like neem, turmeric, and mangista, and we have assays where we can look at how much lipids, we have the sebocytes isolated. We can ask the question, can we actually reduce the lipid production? Because the lipid production is thought to be one of the factors that promotes some of these bacteria. And so this is actually very impressive results. We found that turmeric, manjishta, and neem extract, just aqueous extract, we're not even getting fancy extracts here, really inhibited this. Now, if you took some of the active components like the terpenoids, which are supposed to be immunomodulatory in neem, azadirachtin, or curcumin in turmeric, they didn't have the same effect. So there's still something else in these plants that is doing this effect, and this is one of the areas I was hoping that we could come with some sort of way to collaborate. So we can look at some of these plants, modulate some of these plants, and maybe even in culture, look at some of these plants to see if we can come up with a new way of looking at acne. Now, in general, when you expose these sebocytes to Propionibacterium acnes, this is the bacteria, when you expose them to the bacteria, they actually will produce more lipids. This is already known. But the question was, if we expose them to this turmeric, manjishta, neem, along with the bacteria, can you inhibit that effect? And in fact, that's exactly what happened. So not only are we seeing the effect just in culture, we're also seeing the effect under the conditions where the cell would be in the normal human body. Now, these are not live bacteria, these are heat-killed bacteria, but we know that heat-killed bacteria can still stimulate the cells. Not only that, we looked at the genetic pathways for lipid modulation, and we found, yes, even the lipid enzymes were inhibited with these turmeric and with neem. And I don't show manjishta here, but manjishta too. I haven't been talking about manjishta much because that's something that I didn't want to discuss too many conferences till we actually understand better. Because manjishta has the most promise of all of these, because it also inhibits the proliferation of these sebocytes. So there's actually a lot of promise in this plant, potentially. This is San Francisco. If any of you have had a chance to come visit, it is one of the most beautiful skylines that you'll see. This bridge is the Bay Bridge, not the Golden Gate, but it's a beautiful bridge as well. So when you come, we're only about an hour away from here, so you'll probably enjoy this very much, so please come visit. Finally, this is the area that I wanted to, in the last 10 minutes, just talk about some of the interesting ways that I was hoping we could approach. Because we did some of these studies, but now I want to get a better idea of how can we actually understand the mechanisms and try to enhance the medicinal qualities of these plants. Before we go there, this is interesting. So Ayurvedic medicine, if you look at the Ayurveda practitioners, they always tell you everything starts with the food and nutrition and the gut. They talk about pitta in the digestive tract and everything about the digestive tract. Well, lo and behold, in 2013, modulating the human gut microbiome as an emerging therapeutic paradigm. So their whole point is, if we can control how the gut's health is, we can actually control chronic disease elsewhere, including diabetes, including heart disease, including hypertension. The idea is, in the gut, if you can control the bacteria there, it releases less inflammatory mediators into the rest of the body. So from a Western perspective, everyone's saying, oh my God, look at this is amazing. The Ayurvedic practitioners are saying, wait, we knew this for 1,000 years. What's so great about this? So it's just one of those things where we're finally, the Western science is coming up to it. One of the areas that I find very interesting is how can we stress plants to then enhance their medicinal qualities. And this is an area that's actually been studied. And, you can expose them to UV light, you can wound them, you can expose them to pathogens. You can do, I think, Dr. Streeter, we were talking about nitrogen content in the soil. Sometimes they'll artificially lower the nitrogen content to then enhance the phytochemicals as well. You have to be careful, of course. Anytime you stress the plant, you can overstress the plant and kill the plant, and then you don't get any phytochemicals. So it has to be a balance. One of the areas that we're really interested in is, can we take human pathogens, expose them to the plant, and can the plant then respond to the human pathogen to form certain phytochemicals? And then can that be used then to treat the disease that was caused by that human pathogen? So what I mean is, can we expose these to maybe something like heat-killed Staph aureus? When eczema, Staph aureus is high. Will that induce the plant to produce certain phytochemicals against Staph aureus, thereby making it that much more of an effective plant to treat eczema? Nobody knows. Nobody's ever done these studies. They've only looked at plant pathogens. But there are a few pathogens that cross over between humans and plants, and one is Pseudomonas. The Pseudomonas entire species, there is infectivity in plants and in humans. And there may be other ones that I'm not aware, and this is something I wanted to talk about potentially. So sometimes, here's another piece of literature I wanted to show you. In lettuce apparently respond to many kinds of stress and increase their phytochemical content. Here what they did was they did regulated water deficits. So when they took away the water, the amount of phenolic content actually rises during the period of starvation. But then when you rewater the plant, it'll go back to a level that was lower than the original. So it's almost like you should be harvesting the plants after water deprivation. The farmers know this in the States. What they do is right before they harvest, they will deprive the plants of water for a couple days, and then they'll go pick everything. And their rationale is their fruits taste better. But of course, that means also the phytochemicals are being enhanced because taste is partly due to phytochemicals as well. And what they show is every time you do a water deprivation, you get a slight increase in the phenolic content. The idea is, can we do this same sort of a principle with some of these medicinal plants, so thereby creating an enhanced medicinal yield from these plants. Finally, so that was one area of collaboration that I was thinking about. Finally, one of the other things is, can we take skin and talk about general health? So sebocytes and adipocytes are the fat cells inside, whereas these are oil-producing cells on the skin. So I will tell you, both are lipid-secreting cells. Both have this lipoprotein receptors. And they both have PPAR gamma receptors. And they both respond in very similar ways. In fact, some people believe when you have high cholesterol in the body, the sebocytes, what they do, what we've shown, I'll show you right here, when you have a lot of lipoprotein, they actually increase their lipid synthesis. And one of the hypotheses that's been put forth is if you have a lot of lipids in the body, your skin is trying to get rid of them by excreting it through the sebocytes. So can we use that same principle to then, can we somehow look at the sebocyte character on the skin to then back calculate what your lipid status? That would be nice. It's kind of a dream, but it would be nice if you can swab somebody's skin and predict their cardiac risk. So that would be obviously something that would be very interesting. So sebocytes, what they do is when they produce the lipids, they spit off an entire portion of their internal contents. Unlike adipocytes, sebocytes will what's called holocrine secretion. They'll pinch off an entire portion of their cell and they just spit it out. So whatever you see on the surface is actually what's inside the cell itself. So the idea is, can we measure skin surface characteristics to predict internal lipoprotein status? Other way of looking at it is, if we find plants that can inhibit this process, are those plants also going to be useful in adipocytes? So can we use sebocytes as a first pass way of understanding potential therapeutic targets for obesity? So that's one area that I'm collaborating with a USDA chemist who's very interested in adipocytes. After I showed him the data on the sebocytes, they became very interested. We're putting in some grants and looking at how we can put this together. What we found is when you expose them to this lipoprotein, if you expose them to certain extracts like neem, you can actually reduce that synthesis back down. So neem, again, these plants are not just passively doing some anti-inflammatory function. They're actually modulating the lipid synthesis pathway. So again, there's some other bioactive active component that they have. We've shown this in Western blots as well, where with Mangishta, for example, you get an elevation, then a decrease when you're actually exposing them to the plant. Now, interestingly, if you do very low density lipoprotein, which is more diet related, high in triglycerides, LDL, which is considered the bad cholesterol, both of these increase the synthesis of lipids. However, the good cholesterol has no effect. We don't understand why, but there's something about these cholesterols where these sebocytes see it and then it really activates them to start spitting out lipid. Whereas the HDL, which is the good cholesterol, keeps them dormant, keeps them from being active. This is a very interesting area. We don't understand why, that we're following up on this to understand. But somehow the body has a smart system in place to deal with internal lipids. Can we capitalize on that with some sort of medicinal therapies to augment what the skin is trying to do? Now the problem is, they've just shown rosacea, which is common in the Indian population. It's this red rash that really comes on the face. They believe it's associated with the sun exposure. There was a brand new article that came out of, I think it was Norway, but they published saying rosacea may be associated with cardiac risk. So people with rosacea, if they're associated with cardiac risk, could it be that when they have higher LDL, they're producing more lipids, and that's what's causing that rash on their face. It's actually quite plausible, and this is an area that's actually pretty interesting because it goes to show dermatological diseases are not just skin diseases. They may be a clue that something else is going on, and if we miss that, then you may get lack of diagnosis for many years, and then only when you get a heart attack do you realize you had some problem. So this is an area where you're thinking about earlier intervention and earlier treatment. So to conclude, I just wanted to give you kind of an overview of all the areas that we're working on. So choosing the extract is very important, and then you have to pay careful attention to the solvent. There are a few things that reduce lipid synthesis. We're only touching the surface, and they seem to have greater effect than currently what is thought to be the phytochemicals that are active, which means we don't fully understand all of what's in these medicinal plants, which means it's an area that we can actually go after for more study. And finally, the study of sebocytes may be a really interesting way to look at not only skin surface biology and problems, but you talk about internal lipid status, internal disease, then you're starting to make that collaboration from external disease to internal disease. From an Ayurvedic principle, this is already one thought is anything you see on the skin is already a reflection of your internal status. And in dermatology, we know this to be true. A lot of people will come and say, oh, it's dermatological, it's skin disease, it's not a big deal. They say that until we diagnose them with something where now they have to go see their doctor and they have to make sure they take care of their internal heart disease or their diabetes. There are different clues we can see on their skin where we've caught diabetes more than one time, we've caught heart disease more than one time, something that they didn't know was actually related to an internal problem. So the connection between nutrition and the skin is very interesting. One of the areas that I think would be very interesting is when we're treating these skin diseases, we're giving antibiotics. If we can do some nutritionally augmented intervention instead, either through medicinal plants or even something like millets or something where we can give them something that we can then follow in the skin, I think that's a very huge contribution when you think about general preventative health, so that you're not overloading the system with people that are showing up with heart attacks. Instead, they take an active, engaged approach to their lifestyle. So that's the idea behind it. Thank you very much. It's been a pleasure to come here and speak to you all. These are a lot of my collaborators. As you can see, some are in the wine industry over there. They're excellent, excellent analytical chemists, plant scientists. The College of Ayurveda is on board. We have some sebocyte biologists who serve as my mentor, and then some cell biologists. And this is only a partial list because I'm only showing you partially what we're doing. There's a whole group of other people that do things on Food for Health Institute. Li Tian, for example, is a geneticist. She likes to insert genes into plants to overproduce certain phytochemicals. I think you're working on that with the rice, I believe. And so these are areas that I think there's so many areas that we can move forward in. But so I wanted to give you an overview and say, I'm not coming as a physician to tell you how to eat. I'm actually coming to see if you can tell me how to eat. So thank you very much.